Hello, dear student. I welcome you all on behalf of Sri Jayendra Rank Zone classes. And we are in a continuation of the chapter that is molecular basis of inheritance. Dear students, in some of the previous classes of this chapter, we have already concluded or we have already studied about the different experiment which is carried out in the search of genetic material, structure of genetic material, and central dogma, reverse central dogma. And then we have also discovered the particular one important process or mechanism and the experiments, experimental proof of that, that is a replication. Today, we are going to talk in this lecture about transcription. Dear students, if we talk about transcription, what this term states? This is a term which explains that there is a formation of mRNA from the DNA. So DNA to mRNA formation is known as transcription. So if we talk about transcription, it is the process of copying of genetic information from one strand of DNA into RNA. Means whatever the genetic information which is stored in DNA that is copied or that is transferred into the RNA, then it is known as transcription. So dear students, this is the DNA strands, and out of these two DNA strands, by the use of some enzymes, the single stranded molecule of RNA is getting synthesized. So in this, what happens? The DNA is a double stranded. So out of two strands, one strand is considered as a template, and the mRNA are synthesized or RNA is synthesized complementary to that strand. Suppose the DNA strand is 3' to 5' end, that RNA synthesis occur in 5' to 3' direction, or ribonucleotide are joined in a 5' to 3' direction. Okay, and this 5' to 3' direction is always found. Okay, so this way, if we talk, the transcription take place, and this is the enzymatic process. So here the principle of complementarity governs the process of transcription. Process of complementarity or a principle of complementarity means if the DNA strand is 3' to 5', then whatever the ribonucleotide are going to add in the RNA in 5' to 3' direction are complementary to that DNA strand which is in a 3' to 5' direction. So here complementarity governs the process of transcription, except that the adenosine now base pair with the uracil instead of thymine, because in ribonucleotide thymine is not going to be added because thymine is added in DNA only, but uracil is added in case of the RNA. And here you can see that thymine is pairing with the adenine in case of the DNA. But if we talk about a guanine in front of cytosine come in DNA in front of T, A is going to take a position in cross in plus of A. U is going to take a position in case of uh, particularly in RNA. In RNA in front of A, uracil is going to be added. If in DNA there is a, then a uracil is going to be added in the RNA, and you can see in this particular pictorial arrangement which is shown here. And here you can see this, the uracil are added in front of the adenine of the DNA in RNA. The red color line is showing nothing but RNA. However, unlike in the process of replication in which one sets in, however, unlike the process of replication in which once sets in, the total DNA of organism get duplicated. Means in a one go, whole DNA is going to be duplicated. But in transcription, only a segment of DNA is going to be transcribed, and only a one strand or is copied into the RNA. So if this is the DNA, you can see if this is the DNA, and if this is the DNA and it is a particularly 3' to 5' direction, and it is a 5' to 3' direction, so this 3' to 5' is going to be added template, and the RNA is going to synthesize in the 5' to 3' direction by the RNA polymerase enzyme. The enzyme which is going to be used, that is nothing but RNA polymerase, that is going to form a polymer of ribonucleotide, that is nothing but the RNA. Okay, so here they are going to use a DNA and going to form RNA. So here This RNA polymerase is nothing but DNA dependent, DNA dependent RNA polymerase is added. Firstly, in both strands act as a template. Now the question is, why both strands are not going to be act as a template? Only we are using a one strand of DNA and which is acting as a template, and complementary to that a RNA strand is getting synthesized. Why it is such that both strands cannot transcribe their own different RNA? On, or we can say a question is like that, why only one strand acting as a template? So firstly, if both strands act as a template, they would code for RNA molecule with a different sequences. If both DNA strands will act as a template, they are going to form a RNA with the two different sequences, because the complementarity does not mean being identical. Complementary means it is going to be add complementary to whatever the nitrogenous nucleotide is present on the DNA. And in turn, if they code for if these two different RNA going to code for a protein, the two different protein with the separate or sequence of amino acid in the protein would be obtained. Hence, one segment of DNA would be coding for two different proteins, and this would complement the genetic information transfer machinery. Because if two proteins are getting from the particularly one strand, then it is somewhat two different protein are produced by the same segment of DNA. That is, it is going to complicate the machinery. So this is the first reason why both the strands are not copied during the transcription. Secondly, if two RNA molecule if produced simultaneously, they are complementary to each other. So they can form different protein, but they are complementary to each other, so they can bind. And if they bind, there will be a production of double-stranded RNA. And this double-stranded RNA would prevent RNA from being translated into protein. So the translation will not occur, and protein will not get synthesized if they get a double-stranded. And the exercise of transcription would become a futile one. Transcription will not occur properly. So to understand this 2 point, suppose this is the DNA strand. If this is the DNA strand which having A T G C C G T A C, okay, and this is a 5 dash. This is a 5 dash and T A C G G C A T G, and this is 3 dash. This is the two strands. Now when these two strands act as a template, this is A T G C C G T A C, okay. If this act as a template, then its RNA is going to be 5 dash in front of adenine, the uracil will be added. Thymine is not going to be added. So A C G G C A U 3 dash. Now this is the this template strand. Now if we take this as a template, now synthesis of mRNA take place in the 5 dash to 3 dash direction. Now this is a 5 dash to 3 dash, and this is a 5 dash to 3 dash. This both are complementary to each other. These both are complementary to each other. If they are both are complementary to each other, if they unite, if they unite, they are going to form a So these are complementary to each other, and if they bind with each other, they are not going to synthesize a protein. And if they code for a protein, then the amino acid which they will code, that is different from this mRNA. And if this is the mRNA, this is a mRNA, and this is also the mRNA. So if this will code, this 3 nucleotide will code for, suppose, amino acid 4, amino acid 5, and amino acid 6. This both amino acid are different in a polypeptide chain. So these two different polypeptide will be obtained. So because of this, the particularly two strands are not going to act as a template. Now, to carry out a transcription or to form a mRNA. So now we are going to talk about a transcription unit and the gene. See, dear student, transcription unit is nothing but a segment of DNA which is going to be transcribed into the mRNA. Okay, so it is the segment that is going to take part in the transcription process. So transcription unit is a DNA. In a DNA is defined primarily by the three regions in the DNA. So this DNA segment which take part in transcription has a three regions in the DNA. One is promoter region, another is a structural gene, and the third one is the terminator. These three sequences are present in the segment of DNA which is going to transcribe into the mRNA. There is a convention in defining the two strands of DNA in the structural gene of a transcription unit. So terminology, convention means here terminology are getting used. So this is a schematic structural diagram of a transcription unit. This is the segment of DNA. One strand is in 3' to 5' and another strand is in 5' to 3' direction. See, this 3' to 5' direction is known as template strand because mRNA synthesis always occur in 5' to 3' direction, and mRNA is going to be complementary to this template strand. So this template strand is known as non-coding strand. It is known as non-coding strand or nonsense strand, whereas this coding strand is known as non-template strand or sense strand, because the template mRNA is going to be complementary to this template. So this coding strand is also complementary to this template. So the sequence in mRNA is going to be somewhat similar to the which one? Coding strand. Yes or no? Because this coding strand is also complementary to this template strand, and mRNA is going to be complementary to this template. So it is known as coding strand. Why? Because the sequence in this 5' to 3' direction in a coding strand is almost similar to the mRNA. So it is coding. So it is preferred that this information is transformed in the mRNA through the template. Now, apart from this on template strand, you will see the initial region on this side that is known as 3' or 5' region, that is known as promoter. And this is the transcription start site from where the transcription starts. This is the promoter region. And here, this is the terminator region where the transcription terminates. So from promoter, the transcription initiates. This is the structural gene which are going to be transcribed. And this is the terminator where the transcription ends. Okay. So this is the non-template strand or sense strand. Now, since the two strands have an opposite polarity and the DNA-dependent RNA polymerase, that is a transcriptase, catalyzes the polymerization only in one direction, that is 5' to 3'. So the strand that has polarity 3' to 5' act as a template, and it also referred as a template strand. So this DNA-dependent RNA polymerase is going to synthesize mRNA complementary to the template strand. So suppose this is the template strand. So here this is the mRNA which is getting synthesized in 5' to 3' direction. So if this is the template strand which having a GACTA sequence, Then the RNA strand obviously have CUGAU strand that is in the 5' to 3' direction. The other strand which has a polarity 5' to 3' in the DNA, and the sequence which is on this strand is same as RNA except thymine in DNA at place of the uracil in RNA is displaced during a transcription. So this is the coding strand which has a sequence similarity with the RNA with this exception. So if we talk about this strand is acting as a template strand of DNA, and this is acting as a coding strand or non-template strand. So you can see if the this RNA polymerase synthesized in 5' to 3' direction, it is adding AUCGU in complementary to this DNA sequences and G. Now if you see this sequence on mRNA, this is similar to this. C. The A is there only in place of U there is a thymine. C is there, G is there, U is there, and G is there. So this sequence is similar to the mRNA. So because of this, it is known as coding strand. Strangely, this strand which does not code for anything is referred as actually it is not coding for anything. Then also it is referred as coding strand because there is a sequence similarity with the RNA. Okay, so this is a coding strand, but it has a sequence similarity with the RNA. So all points while defining the transcription unit are made with reference to the coding strands. See, remember all the things: promoter, terminator, structural gene, all things are described with a reference to the coding strand. And coding strand have a 5' to 3' polarity. So to explain the point, the hypothetical sequence from the transcription unit is represented below. And if this is the hypothetical sequence in the transcription unit in mRNA, in DNA, then it is a template strand and there is a coding strand. So if mRNA gets synthesized, the promoter and terminator flank the structural gene in the transcription unit. Promoter is present at the 5' end to the coding strand, whereas terminator is present on the 3' end of the coding strand because these all are defined based on the coding strand. So the promoter is said to be located towards the 5' end that is upstream. Now see, remember, the 5' end of a coding strand is represented as upstream and 3' end of a coding strand is represented as a downstream. So promoter is located towards the 5' end that is the upstream of the structural gene. The reference is made with respect to the polarity of coding strand, and it is a DNA sequence. Promoter is nothing but a DNA sequence that provides the binding site for the RNA polymerase and from where the transcription initiates. So DNA sequences are present in the promoter that provide a binding site for RNA polymerase, and it is the presence of the promoter in the transcription unit that also defines the template and coding strand. That also defines the template and coding strand. That 5' end has a coding strand and the 3' end strand is the template strand. And if promoter is present on the 5' end of the coding strand and terminator is present on the 3' end of the coding strand, if we switch the position of with that of the terminator or switch the position of the promoter with the terminator, the definition of the coding strand and template strand will be reversed. Means the strand which is acting as a coding strand now will be acting as a what, a template strand, and the template strand will act as a coding strand because the position of promoter and terminator will be switched. Terminator, if we talk, terminator is located towards the 3' end. Obviously, as we have seen in the diagram, it is located towards the 3' end, that is the downstream of the coding strand, and it usually defines the end of the process of transcription. Means it is the point where the transcription completes. There are additional regulatory sequences. They may be present further upstream and downstream of the promoter. Okay, further additional sequences are also found. Now, what are these additional sequences? That first we need to discuss. Then we will discuss about some of the questions. So now, as you can see in this diagram, this is the coding strand and this is the 5' end. So promoter is present towards the 5' end of the coding strand, and terminator is present at the 3' end of the coding strand. And in between, these are the structural genes which are coding for the enzymes, and this is the site from where the transcription initiate. Now, in this promoter, there are different sequences which are actually recognized by the RNA polymerase enzyme or the enzymes. So if we talk about the sequences, then in prokaryotes, dear students, recognition sequences recognition sequence is present in promoter region. Present in promoter region at upstream or towards 5' end of coding strand. And this recognition sequences for this recognition sequences are these recognition sequences are number one, primnomox. Now primnomox, or we call it as a TATA 80. Okay, it is minus 10 sequence. It is minus 10 sequence or 10 base pair upstream ten base pair upstream from the start point. So these sequences are present in promoter region, and that is 10 base pair upstream. Means suppose this is the transcription unit and this is the transcription start site. These are, suppose here the promoter is there. Then between the transcription start site and the promoter, 10 base pair if present, means I can call this promoter region is present 10 base pair upstream. I am going to draw a particular diagram again in a proper way. Okay, so it is a 10 base pair upstream. Another sequence is also there we are going to draw it on a one sequence, that is TTGACA, that is minus 35 sequence, or it is present 35 base pair upstream from start point. Means suppose this is the DNA strand, this is the DNA strand, this is 3' to 5', this is 5' to 3'. Now this is the coding strand, and on coding strand this 10 base pair upstream, 35 base pair upstream is present, that is TTGACA. Okay, and then here TATA80 is present, and here this is transcriptional start site. So if it is a 10 base pair upstream, means between the this minus 10 sequence and the transcription start site 10 base pair are present, and this is present at the minus 35 sequence, means between the start site and the minus 35 sequence 35 base pair are present. Okay, so if between the transcription start site and the minus 35 sequence a 35 base pair are present, means we can say this sequence is 35 base pair upstream. Whereas if TATAAT is 10 base pair upstream, then it is known as a Pribnow box or TATAAT or minus 10 sequence. Okay, so this is seen in case of the prokaryote, but in eukaryote this recognition sequences in promoter are different. So if we talk about that, in eukaryotes, recognition sequences of promoter are: number one, it is Tata box or Hognis box. Now this Tata box or Hognis box is 7 base pair long, and it has a sequence that is T8T8T8T or TATAAAT. And this sequence, which is 7 base pair long, it is known as Tata box. It is present or located 20 base pair upstream to the start site. And another sequence is the cart box, which is present between minus 70 and minus 80 base pair. It is present between minus 70 and minus 80 base pair. Now here, suppose this is the transcription start site. So here you will see that 20 base pair are present between the start site and the Tata box, and it has a sequence that is TATATAT. Okay, whereas if you talk about this cart box, this is present minus 70 to minus 80. So here the C880 sequence is present. This is suppose 70 base pair are present between this, and here from transcription side file 80 base pair are there. So if between 70 and 80 this base pair is present, this particular sequence is found. So it is it is called that it is present between this minus 70 to minus 80 base pair upstream. Okay, so such sequences are found in the particularly we can say the mRNA. Now apart from this, you will see another sequence on the mRNA that is known as Shine-Dalgarno sequence. Shine-Dalgarno, which is also known as SD sequence. If we talk about this Shine-Dalgarno sequence or SD sequence, it is 5 dash AGG AGG U3 dash. It is AGG AGG U3 dash, and this AGG AGG U, if we talk, it is present 5 dash, and this is the sequence which is present at 5 dash and near initiation codon, and initiation codon is nothing but AUG, and it is found in prokaryote only. It is not found in eukaryotic RNA. Okay, it is found in prokaryotic mRNA. And the function of this sanitary Karamu sequence is function, if we talk, then in it help in binding of 30S subunit to 30S subunit of ribosome to prokaryotic mRNA. So we remember this is found in prokaryotic mRNA only. In eukaryotic mRNA, it is not going to be found. Okay, so this is the particularly we can say transcription unit, which is this kind, which is going to form a mRNA. Now after this, now in this transcription unit, the gene which is involved in the formation, the gene segment of DNA or a gene which is known as transcription unit, which is involved in a particular formation of mRNA. So a gene is defined as the functional unit of inheritance. Gene is defined as a functional unit of inheritance that is involved in mainly inheritance. Now, though there is no ambiguity that the genes are located on a DNA, genes are located on DNA. So DNA is getting transferred from generation to generation, and such a way the DNA gene also getting transferred. So it is difficult to literally define a gene in terms of DNA sequence. It is difficult to literally define a gene in terms of DNA sequence. So suppose this is a segment of DNA, chromosome is there. If chromosome decondensed, the DNA will be obtained and the segment of DNA will be considered as a gene. So the DNA sequence coding for tRNA or rRNA molecule also defines a gene. So a gene is nothing but what? Which can code for any types of RNA. And further, it will be coding for the, ultimately this RNA will be coding for the particular proteins. But it can code for tRNA, it can for RNA, and if it code for the mRNA, it is going to form a protein. The structural gene in a transcription unit which we have studied that can be said to be monocystronic or polycystronic. In monocystronic, they are mostly in case of eukaryote, but in mostly in case of bacteria or prokaryote, this particular structural gene in transcription unit is said to be polycystronic. So structural gene in transcription unit is either monocystronic or polycystronic. Monocystronic mostly in eukaryote and polycystronic mostly in bacteria or prokaryote. And if we talk about cistron, cistron is nothing but it is a segment of DNA which code for a polypeptide. It is a segment of DNA which code for a polypeptide. So if we talk about a eukaryotic DNA, it is monocystronic means as you can see here, this is mRNA, and this mRNA structural gene which code for a one protein. So if it is known as monocystronic structural gene, then they are going to code for only mRNA. That mRNA going to code for only one protein. So if the gene or a structural gene code for a one mRNA and that one mRNA completely code for a one protein only or a one polypeptide only, then it is known as monocystronic. So in eukaryotes, the monocystronic structural gene have interrupted coding sequences, and the genes in eukaryotes are split. Okay, so gene in eukaryote have some of the coding sequences and some of the non-coding sequences we will see. But if this is the mRNA which is synthesized from the DNA, this is the mRNA, and this is particularly coding for the one protein only, and it is a monocystronic. Now, the coding sequences or express sequences are defined as exons. Means in eukaryotic mRNA, if you will see some of the coding sequences and some of the non-coding sequences. Coding sequences are known as exon. So exons are said to be those sequences that appear in matured or processed RNA, means because they are important. But the exons are interrupted by the introns, and introns are the non-coding sequences. And introns are not going to be found in the processed RNA or a matured RNA. Okay, so if this is the DNA, you will see that this particularly are the exons. Okay, these are the exons, and in between you will see the introns. Introns are the non-coding sequences, and if they are non-coding sequencing, that has to be removed, and that are removed after transcription in a post-transcriptional modification. In case of eukaryote, we will study this post-transcriptional modification after studying the transcription in eukaryote. Now, introns or intervening sequences do not appear in matured or processed RNA, and they are removed. So, like this, you can see this blue part is getting removed after the post-transcriptional modification. That is the elimination of intron. Okay, in transcript signal, and the splicing of exon occurs. Splicing means removal of this intron, it is known as, and joining of this exon, it is known as splicing. And this is specifically found in case of eukaryotic DNA. Please remember. So this split gene arrangement means the presence of intron in between the exon further complicates the definition of a gene in terms of the DNA segment. Okay, because gene code for a polypeptide, and all particular segment in DNA are not able to code for a polypeptide. So inheritance of a character is also affected by a promoter and regulatory sequences of a structural gene. Hence, sometimes the regulatory sequences are loosely defined as regulatory genes. Means we can call a promoter. Okay, inheritance of a character is also affected by promoter and regulatory sequences of a structural gene, because structurally it's going to be processed in the mRNA, but this promoter and regulatory sequence are not going to be transcribed in mRNA. So sometimes the regulatory sequence are loosely defined as a regulatory genes. They can be called as regulatory genes, but they are not going to be transcribed in the mRNA in a mRNA of a structural gene. So even though these sequences do not code for any RNA or a protein, these regulatory sequences, they are particularly, we can say, known as regulatory sequences. So if we want to write a definition of monocistronic and polycistronic mRNA, then monocistronic mRNA is the mRNA which code for only one polypeptide. But if it is a polycistronic mRNA, means it has a information of more than one polypeptide. So this mRNA code for more than one polypeptide. And if this occurs, then this monocistronic mRNA, as we have studied, it is found in eukaryotes, tick cell or eukaryotes, and this polycistronic mRNA is found in prokaryotes. Okay, so such a way this can be defined. Now, if we want to continue further, then we are going to, now after discussion of structural gene and or we can say a transcription unit completely which is going to code for the mRNA, now we are going to talk about the different types of RNA. And if we talk about the types of RNA in bacteria, there are three major types of RNA. If we talk, there are messenger RNA which are transcribed from the mRNA and going to form a protein. There is a tRNA which are known as transfer RNA, and there are RNA which are known as ribosomal RNA. All three RNA are needed to synthesize a protein in a cell. There is a need of all these three types of RNA. Because when we will study translation, I will describe you which mRNA is used where. mRNA, if we talk messenger RNA, tRNA, and rRNA, the messenger RNA provides the template. It provides the template for the synthesis of protein. tRNA reads the genetic code, means it reads the mRNA genetic code present on the mRNA, and genetic code. What is genetic code? We are going to discuss in the next lecture, and bring specific amino acid. So, what is the role of this tRNA? To read the code on the mRNA, and by reading the code on the mRNA, provide a appropriate amino acid, or take appropriate amino acid and join in the polypeptide. Whereas if you talk about rRNA plays a structural and catalytic role during the translation. So it will be acting as an enzyme, and it has a mainly role as a peptide bond formation, and it is also a part of ribosome. So if we talk about all this RNA in detail, then so let's discuss first mRNA, and this mRNA is also known as messenger RNA. This is also known as messenger RNA. Now, if we talk about this messenger RNA detail, that is a long RNA molecule. It is a long RNA molecule that constitute 2 to 5% of the 2 to 5% of total RNA content. Means whatever the amount, whatever the different type of RNA is present within the cell, out of a total 100% RNA, 2 to 5% RNA are going to be of mRNA, not more than that. And it is known as messenger RNA because it brings instruction or information from the DNA for the formation of a particular polypeptide. So it is known as messenger RNA because it brings information from DNA. It brings information from DNA for formation of particular polypeptide. Therefore, it is also called as informational or genetic RNA. Informational RNA or genetic RNA. Now, the information for polypeptide is present in form of nucleotide sequences called genetic code, and three adjacent nucleotide on mRNA code for one particular amino acid. Okay, so that is the thing. Means this ribonucleotide sequence is present on the mRNA, which is complementary to the template strand on the DNA. But the three adjacent nucleotide code for a one amino acid, and that sequence is known as genetic code. And this formation of polypeptide from the mRNA is done by the ribosome. Okay, so this is the thing occurs. Now, in eukaryotic, if we say the modification occur in eukaryote, what happens? The particularly modification occur in mRNA and different speaker sequences are found on the mRNA. So if we see the structure of mRNA, then in mRNA you will see the presence of 5' cap. Then apart from 5' cap, you will see the sequences. And here these are acting as a initiation codon. And initiation codon is AUG. Now this is the coding region for polypeptide. And here there is a termination codon where polypeptide formation or protein formation stops. And here you will see the poly A tail. So how this poly A tail is added, how this 5' cap is added, that all we will discuss in the post transcription modification where we will study the after transcription in eukaryotes. These are non-coding region in mRNA, and this non-coding region are known as leading region. These are known as leading region, whereas this sequences are known as non-coding region, or we can say tail region. Okay, so these are the parts of mRNA strand. Which we have discussed in this particular diagram of mRNA. Okay, this is the diagram of mRNA strand. Now in this, cap is present at before the initiation codon at a 5' end. Now the small non-coding region is present, leading region is present between the 5' cap and the initiation codon. Okay. Now that is a coding region followed by a termination codon. This termination codon can be UAA or UAG or UGA. Out of three, any one can be present. And after termination codon, there is a small non-coding trailer region or a tail region. And the leader and trailer region are called UTR. See, this is also known as UTR, and this is also known as, sorry, this is also known as UTR. So what is UTR? UTR are untranslated region. They are the untranslated region. And this untranslated region are known as UTR sequences. So this UTR are untranslated region which are not getting translated into polypeptide, but these are present before start codon at 5 dash end and after start codon at 3 dash end. Okay, so this is the what particularly one. This is the longest RNA. Okay, so this is the thing. Now mRNA, it is longest RNA, and this mRNA, it is called template. It is called template or nuclear or messenger or informational RNA. Lifetime is also variable. Lifetime can be from few minutes to few hours. On the other hand, mRNA of the higher form can have a long lives that can be few hours. Lifetime is of several days in case of young, RBC, which continue to form hemoglobin even when nucleus has degenerated, which continue to form hemoglobin even when nucleus has degenerated. Okay, so this is the thing which found in case of the particularly mRNA. Now after mRNA we are going to talk about the tRNA. Okay, that is a transfer molecule. So now we are going to talk about the second type of RNA, that is tRNA, which is also called as transfer RNA. हाँ हाँ ठीक है हाँ. This three-dimensional structure of tRNA, if we talk, it has a L-shaped form. Okay, so dear student, till now what we have discussed? In the previous slide, we have discussed that they have a particularly different acceptor arm where is the 3 dash end where amino acid binds. Then they have this loop and this is the stem, DHU stem, D loop. D loop has a 8 to 12 bases. It is largest loop and it is amino acids, and that is enzyme binding site. Enzyme binds here. This is the DHU arm, so it is DHU stands for dihydroxyuridine. Whereas this fourth loop is a T-Psi-C loop because Psi is a pseudo-uridine. Bases are here. This is T-Psi-C stem and loop combinedly form a arm. Now, if we talk about this T-Psi-C loop, that is a 7 bases long and it is a ribosome binding site. Now, anticodon stem is there and anticodon loop is there. It is an anticodon and it is an arm. Now, this anticodon is also the nodok, and this anticodon has a complementary sequences to the mRNA. Okay, so this is the amino acid binding site and this is a variable arm. The function of variable arm is not known. Okay, all the information is written here. Now, what I'm going to say here, see, this is the tRNA. Now this tRNA has a mRNA sequence, and mRNA has a AUG codon, then a complementary sequence is going to be UAC. So AUG code for methionine. So whatever the tRNA which has a complementary sequence, that is a UAC, it has a methionine. Now if we talk about 2D structure and 3D structure, 2D structure is a clover leaf like, and 3D structure is L-shaped form. 3D structure is L-shaped form. Okay, so this is the particularly structure of tRNA. Now if we talk about the L shaped form, that is a 3D structure, if we talk, then the L shaped structure is somewhat like this. So let's draw it in the next slide. The L shaped 3D structure of tRNA. So this way the structure is formed. Now in this there is a somewhat bonding like this between the complementarities there. Okay, this is a 5 dash N, this is a 3 dash N. This is ribosomal binding site. In this ribosomal binding site is D-Cy-C site, whereas this 3 dash N is amino acid binding site. This is enzyme binding site, and this is anticodon loop. Okay, so this way the structure is formed like this. Okay, now after this tRNA structure and the detailed study about the 2D 3D structure, now we are going to talk about the third type of RNA, that is rRNA. It is known as ribosomal RNA. Now if we talk about ribosomal RNA, it constitute it constitute 80% of the total RNA content of cell. It is a smaller RNA then mRNA, but longer than the tRNA. And if we talk about the rRNA, it is the most abundant RNA. Why it is known as most abundant? Because 80% it constitute. Okay. Now this is the constituent of ribosome, or is a structural molecule of ribosome. Because ribosome are made up of rRNA and some protein. And if we talk it is made up of rRNA and protein, then if we talk about a 70S ribosome, that this is a large subunit and this is a small subunit. This is 70 as in this is a 50S subunit of ribosome, and this is 30S subunit of ribosome. Now in the 50S subunit, you will see that there is a 23S rRNA. Okay, 23S rRNA, and 16S rRNA are present in this. Whereas 5S rRNA is also present in the large subunit. Whereas if we talk about 80S ribosome, which has also a large subunit and which has also small subunit, this is 60S large subunit, this is 40S. This 40 has as 18S rRNA, whereas this 60S has a 28S rRNA, 5S rRNA, and 5.8S rRNA. Okay, this different type of rRNA are found. So this is the rRNA, which is a structural molecule of RNA, and in this 23A and 28S, 23S rRNA in prokaryote and 28S rRNA in eukaryote have peptidyl transferase activity during translation or protein synthesis. Peptidyl transferase activity means transfer of peptide, and that what is that we will discuss in. So this is a catalytic activity, means they have catalytic role. Okay, dear students. So this is about the rRNA. After rRNA, there are other different types of RNA are there. If we talk about other RNA, there in this, first we talk about genomic RNA. If we talk about the genomic RNA, it is also known as genetic RNA. So genomic RNA is the RNA when act as genetic material, then it is known as genetic RNA. And this is found in present in some viruses. After this, the another one is the small nuclear RNA, which are also known as snRNA. These are the small size RNA small sized RNA present in nucleus, and each RNA is combined with 7 to 8 molecules of protein to form a small nuclear ribonucleoprotein. It is combined with 7 to 8 molecule of proteins to form small nuclear ribonucleoprotein or snRNP. Then after this, third type of RNA, that is small cytoplasmic RNA or scRNA. And this small cytoplasmic RNA or scRNA, if we talk, it is a small size RNA occur free in cytoplasm. Okay, occur free in cytoplasm. Then it is known as small cytoplasmic RNA. And this small cytoplasmic RNA help in binding a ribosome to endoplasmic reticulum for producing secretory proteins. Okay. Then the another one is microRNA or miRNA. These are 21 to 22 base pair long RNA, and this attach to complementary part of mRNA and bring about their degeneration. Okay, so this is the particularly some different type of RNA which are found other than the mRNA, tRNA, and rRNA. Okay, I hope you understood this topic, that is about the different type of RNA. So till now we have discussed about the transcription unit, the gene, and the different type of RNA. Now in the next class we will be discussing about the process of transcription in prokaryote and eukaryote. Thank you.